Hello everyone. Thank you very much for coming to this talk in your busy day. I'm Ramesh Raliya from Washington University in St. Louis. I'm a research associate at Washington University, and I'm interested in smaller particle and their application in biomedical use. Often we heard about smaller particle, they affect the health, but I'm interested to use how we can turn a small size particle into for the beneficial purpose. Today I will talk about how we can use a small nanoparticle made up of gold particle, gold metal, to transport this particle into the brain, and why this is important and how we made this transport of the nanoparticle, what is their mechanism, that all we are going to discuss. So, President is speaking here that we can understand and we can study the particle smaller than atom, but we are not able to fully understand the organ, the human body organ, which is less than a couple of pounds. And its importance, the President is speaking in a White House Brain Initiative. The White House government invested a lot of money. The reason behind that, because the brain has thousands of diseases associated with that. And these thousands of diseases, once person get affected with brain related disease, it takes long time to cure. The reason behind that, the picture you are seeing here is that this is your blood and this is your brain area. And in between the blood and brain, there is a strong barrier. And in this strong, we can tell that strong wall between two different regions. The thing is that this is called the blood-brain barrier, made up of several proteins and the lipid molecules. And its main function is to protect your brain. If brain hit with something, it protects the brain. But at the same time, when we intentionally want to deliver something there, in case of some disease, it also protects that thing. And your drug reach to the brain, it takes long time to reach the drug to the brain. Because this blood-brain barrier basically prevent your drug or any molecules to cross through that. And since it can prevent a virus, which is like in 10 nanometer or 20 nanometer scale, then it can prevent anything. So it only takes a smaller particle just like glucose or some ions, so that brain keep getting nutrients. So in case of the mental challenges, and when we want to deliver the drug, what are the options available today? So currently, to deliver the drug to the brain, few things are being practiced by the doctor, which include like invasive technique and the non-invasive technique. Invasive technique means they inject something directly, they pierce your blood-brain barrier, and they deliver the drug to the brain. In that case, you can rapidly deliver the drug to the brain. Let's say if there is an accident, brain damages, and you really want to deliver the drug very rapidly to survive the patient, then they directly inject into the brain by crossing the blood-brain barrier. But in normal cases, they use the pharmacologic or the physiologic ways to deliver the drug. In that case, they used like high osmolar solutions, so that your blood-brain barrier temporarily open for a certain period of time, so that your drug can pass through. But all these, both the process has some drawback. Like invasive process, because brain has several nerves, thousands, millions of nerves are there, and if any one of the nerve get affected, maybe your one of the body function get affected. So this is rapidly we can deliver the drug, but same time we have lot of risk. In this process, non-invasive technique, we don't have any risk to dysfunction the body function, but the only problem is it takes long time. the person once went into the coma or get into the coma, it takes weeks and weeks and weeks to revive from that particular disease. So you need some sort of technique that can deliver the drug very rapidly, and that should be non-invasive technique. So currently you can see the complex system that in general when person affected the disease, we inject the particular drug in a body, and this drug is passed through entire circulatory system, and this circulatory system then it goes to the brain, and then it has to cross the blood-brain barrier. So you can see the how difficult network is in a drawing, but in real system it's really more complex system. But here you can see the picture where you can see that this is a oral cavity, the mouth, the section of the mouth, and here from the nose we respire some gases, and if the gases have some sort of any molecules or any particles, these particles also went into the oral cavity. Also in the next slide I will discuss that what kind of particle size affect and where they go. And in the oral cavity, here the one thing is to note that we have the olfactory bulb, and this olfactory bulb had some fibers of olfactory nerves, and this olfactory nerve ending, what they do, they basically recognize the molecule, gaseous molecule, and once they because they have the receptors to receive some molecules, and then those molecules received by these nerve endings, they transported through the axonal transport mechanism. The axonal transport mechanism is basically the moving of the particular transport of any particular gaseous molecule within the neurons. So once these particles transport through the neural transport mechanism, then there is no blood-brain barrier, and these gaseous molecules rapidly reach to the brain, and brain respond accordingly. For example, if we cross someone or pass someone, and if someone has some spray or particular kind of smell, we can easily recognize within a fraction of a second. So we would like to use the same concept, that how we can use this concept. So here is a graph which says that what kind of particle size, if you have a different particle size in the air or in the gaseous molecule, where this particle will go and deposit if this particle taken up through your nose. So the x-axis shows the particle diameter in micrometer, and y-axis shows that deposition fraction, where this particle will go. So the thing is to note that the red colored line one I mentioned is that if your particle is less than 5 nanometer size, then most of the particle, means around 80% of the particle, will deposit in your head airways. Means 80% of particle will get deposited here. But if your particle is larger than that, these particles will pass through, and this particle will go and deposit into the lab. This is a basic diagram in aerosol science and technology. They basically calculate that how the pollution affected health, particularly the PM 2.5 and PM 10. That's the regulation of the Environmental Protection Agency. So they consider this particular figure, that based on this figure they put the regulation of PM 2.5 and PM 10, that where this particle will get deposited. So we would like to use the same concept. We will make the particle less than 5 nanometer, so that all the particle or the maximum of the particle get deposited in the head airways, and this particle received by the nerve endings, and they transport through the neural transfer mechanism. So to realize the hypothesis, we will make a particle which are like 5 nanometer or less than that. Then we have to characterize the particle, because when we make the engineered particle, this particle might be toxic. How this particle, if you have one particle 5 nanometer, but if you can put all these particles in a single solution, and if this particle comes close and they aggregate, so you have one particle of 5 nanometer, but if two particles come close, your effective particle size is 10 nanometer. So you have to characterize your particle that these particles are really stable in a biological solvent or in a vector which you use to deliver this particle or etc. Then to track that, once you deliver this particle, you need to track that where these particles are going. So we functionalize this particle using some fluorescent tag or fluorescent dye, that where these particles are really moving. So we characterize using the fluorescence microscopy. And once you have the functionalized particle, then you have to deliver these particles. So you need some sort of technique which can effectively deliver these particles, which can generate some aerosol. And aerosol is the particle suspended in the gas or in air. Then once you deliver this particle, then things come that how much particle you delivered and how much particle actually reached to the brain. So you need to do some surgery, and then eventually you have to detect the biodistribution and biointerface, that when the particle reached to the brain, how much particle reached quantitatively in milligram, microgram, ppb or ppm level, and then the bio interaction, that these particles reach to the brain, but how these particles are affecting other body function. Because earlier I told you that when we do the invasive technique, in invasive technique, because brain has lot of nerves and these particles go and they affect the another nerves, then they may affect the another body function. So we don't want that. So we have to study the bio interaction. So let's go step by step. So the first is synthesis of nanomaterial. And here we are synthesizing the gold nanomaterial. We synthesize the gold nanomaterial using the seed mediated approach. Seed mediated approach means first we make the particle of 0.1 nanometer or 0.2 nanometer, I mean very small particle, and then we grow this particle in a growth solution which has some other precursor salt of the gold. So from 0.1 nanometer we grow this particle up to the 5.2 nanometer. So here we grow all these particles in 5.2 nanometer. These are the transmission electron microscope image. One is the little bigger and another is zoomed image. One thing to note from these images is that all the particles you are seeing here, they have a very similar size. And in terms of the colloidal stability, there is an index, polydispersity index. Polydispersity index basically tells that how your particle is stable in a particular colloidal system or in a colloidal solution. So here you can see that polydispersity index of all these particles are 0.022, and the polydispersity index close to 0.001 is considered as highly stable particle, and polydispersity index close to 1 considered as non-stable solution. Means when you put the particle, let's say you put the sand in a water glass, and all sand get and deposit at the bottom. So that polydispersity index is 1. So it says that all the particles are highly stable and they have monodispersion size. Most of 99% of these particles are 5.2 nanometer. So you can keep all these particles for a long period of time. And for the biomedical application, you need a drug, you need a therapeutic agent which can be stable for a long period of time and in a wide range of temperature, like from room temperature to 20 degree, 30 degree, 40 degree. So you need a wide range of temperature. So here all these particles we measured and we kept at the room temperature, and these particles are highly stable. Then once we made these particles, then the thing is came that how to functionalize these particles so that we can track them. So for that we need to know the surface property of the particle. So we need to know what is the surface charge of the particle. So we measured this the charge of the particle, which I didn't mention here. So all these particles are positively charged on the surface. And why we put all the particles are positively charged? The one we can easily functionalize using some agent, which I am going to tell. And the another thing, our biological membrane has a negative potential, -60 millivolt. So if we have any positively charged particle, it can easily attract with a negatively charged surface. So here we have the gold particle, and then we use the cysteine amino acid. Cysteine is one of the amino acid out of the 20 amino acid essential for the human. And what is cysteine? The beauty of cysteine amino acid is, you can see at the top there is a SH group that's called the thiol group, and thiol group has property to bind with the gold nanoparticle surface. And once that thiol group bind on the gold nanoparticle surface, we can attach a dye molecule, a fluorescent dye tag, that's fluorescent isothiocyanate, FITC. So we attach the fluorescent isothiocyanate so that this particle can be functionalized. So here in this picture, you can see that step by step we measured this particle under the fluorescence microscopy from stage 0 to after functionalization. And you can see that with the functionalization, all the particle, you can see the fluorescence from all the particles. And at the same time, to verify the functionalization steps, we did the UV-VIS absorbance measurement. And here you can see that with the when we put the cysteine amino acid or the gold surface, the peak is shifted because the original peak is 524 and then peak is shifted to 530, and then once we put the fluorescent isothiocyanate dye, this peak is again shifted to 482 nanometer. So all these characterization data confirm that the particle when we synthesized, they are functionalized particle. And once we have the functionalized particle, then the question comes that how to deliver this particle. And as I mentioned that we need a technique which can deliver the particle, single particle at a time. Because you don't want to deliver the two particles together, because if particle is 10 nanometer, the particle will start to go into your lung, because those particles will not stay in your oral cavity, and these particles may not have a desired property which we want. So here we choose a technique that called the electrospray. Electrospray is basically a technique which generates an aerosol particle under the electric field. And using the electric field, we can actually tune the cone of the spray. And this particular cone is called the Teller-Conjet mode. And under the Teller-Conjet mode, this spray generates a single droplet at a time. One droplet has a single particle. And because of the high charge, and at the same time we also purge the air as a carrier gas. Because of the air and the electric charge, this droplet get evaporated, and these particles eventually get onto the substrate. And here is the substrate is locust antenna. We choose the locust as an animal to test our nanoparticle delivery to the brain. And why we choose the locust? Because the olfactory nerve system and the olfactory nerve organization is quite similar to the human olfactory system organization. The actual system, so here you may feel that all these particles is directly spread over to the brain. So this is basically figure the actual experimental setup was in a way that the nanoparticle will not directly expose to the brain. This particle will only expose to the locust antenna. And at the same time, we also put a probe, a electric probe in the brain of the locust, and we measure the electrophysiology recording. This barcode is basically showing the electrophysiology recording, that how the brain functioning, how the nerve functioning at the time when we are spraying all these particles on them. So once we deliver these particles, then question comes that whether these particles were delivered or not. So at first instance, we characterize through the qualitative techniques, And qualitative techniques is basically the microscopy technique. And here you can see the fluorescence microscopy image. Here the bottom image shows about the brain and the top image of the antenna. This schematic diagram is showing the experimental setup that we put the gold aerosol on the antenna, and then this is the brain we took out and we did the microscopy. And the fluorescent signal coming out from the brain and the antenna, it suggests that gold nanoparticle reached to the brain. The interesting thing in antenna to note that in antenna you can see that certain points are more glowing, certain regions are more glowing than the rest of the region. The reason behind that locust antenna has thousands of sensilla. Sensilla are a small structure over the locust antenna surface, and their main function is to sense the particular kind of smell. So some sensilla is lighten up, whereas others are not. It suggests that there are specific receptors, there may be specific receptors over the locust antenna surface which can uptake the gold nanoparticle. And the bottom one shows that these particles go into the brain, and since it seems that all our brain is blown up, so might be all these particles are distributed throughout the brain. Once we did this qualitative, then we confirmed that once these particles go into the brain, whether still particles are remain separated, or once these particles reach into the brain, they get aggregated, or if there is any changes in their structure. So we did the transmission electron microscopy. So we took out the brain, and then we cut the slices of the brain, 75 nanometer thick slices, and then we did the electron microscopy. And in the transmission electron microscopy of the brain, you can see in the blue circles, there are some small dark dots. Those are basically the gold nanoparticles. Now you will ask a question or raise a question that how can you say that this black dot is a gold nanoparticle? This might be some other bodies in the biological system. So there is a technique that called the electron dispersive X-ray spectroscopy. So that technique can be coupled with the transmission electron microscopy, and then you can tell with the point scan that hey, this particular particle is a gold nanoparticle or not. But here we didn't use the EDX technique, electron dispersive X-ray spectroscopy technique, but we used the same slice or the entire brain, and we did the ICPMS, inductive coupled plasma mass spectroscopy. And inductive coupled plasma mass spectroscopy technique allow us to quantify that how much particle is really reached to the brain. So here you can see the result of the ICPMS. So we did the ICPMS of the antenna which we exposed through the gold nanoparticle. We also used the unexposed antenna as a control, that we did not expose anything, and is there any gold particle or not, because that was the control experiment, that we did not expose gold particle, and then we did with the brain. So you can see the difference is that the exposed antenna is clearly has the higher gold nanoparticle than the unexposed antenna. And then once we expose the brain, this gold nanoparticle is clearly more accumulating in the brain with the time. So we did the experiment with the 3 different time duration, 1 hour, 3 hour, and the 6 hour. And we see that as we increase the exposure time period, the gold nanoparticle accumulation in the brain is increasing. So it suggests that as you increase the exposure time, as you increase the exposure number of particles, these more and more particles get accumulated into the brain. So now question arises that why the unexposed antenna has the gold, why the control has the gold particle? To find out that at the first instance when we did only for 1 hour exposure and we didn't find any significant difference in exposed antenna and unexposed antenna, then we thought that there might be contamination. But when we did the repetitive experiment, we find the same result. Then we start to analyze that what is exactly reason why the locust has the gold particle in their antenna. Then we analyze the food which we provided to the locust to eat while growing the locust in the colony. Like we provide oats, we provide soil on which they keep growing, and also the water. And we find that certain very small amount, at ppb level, parts per billion content, gold is present in these three things, because all the elements are present in a very minute amount in the all these different substances. And with the time, all these organisms like locust, they have a bioaccumulation power. So they start to accumulate some sort of elements in their body. So because of that, we find the gold in the unexposed area. So the next question comes that we delivered this particle, we delivered in a certain amount. If we expose for a certain period of time, we will get certain amount of particle into the brain. But whether these particles are affecting other nerves or not, that called the biointeraction. So here you can see that the experimental setup again, and this is multi-channel electrode, and we measure this multi-channel electrode through a particular kind of recording that called the projection neuron recording. The projection neurons are basically a bunch of neurons at particular neuron node. And here we use the specific order to measure. The specific order is we use the hexanol as a specific order, because when we smell some particular specific kind of smell, there is a particular kind of spike will come. So in locust, hexanol is well studied that when you expose the hexanol of particular concentration, what kind of nerve spikes will come. So here we in the barcode, you can see that we expose the only order without gold nanoparticle, and then through those recording we exported this data, and we are trying to calculate the firing rate. Firing rate is basically the how the nerve responds once the nerve get any molecule or feel anything, then how fast the brain responding. So the top figure, you can see that here we don't have the gold nanoparticle, we just have the order. So this is the control, and as soon as when we expose the hexanol for a 4-second period, you can see the clear spike there. And after the 4 seconds, when we remove the exposure, then it comes again to the normal. And at the bottom, you can see that when you use the gold nanoparticle plus order in the same manner for the 4 seconds with the order. So you can see there is a spike, but the pattern of the spike remains same, which is along with the hexanol. So it suggests that there is no interference with the other neurons, because if there is an interference due to the gold nanoparticle, it should interference at some sort, at some period. Then again we use this firing range, and we calculate the order evoked firing rate in terms of the heart. So you can see that without gold nanoparticle exposure, when we only expose the order, then there is a certain amount of the firing rate, and then when we expose the gold nanoparticle at the beginning, when we start the experiment, and after 2 hours of experiment, we did not find any significant changes in any of these. So it suggests that the particles we synthesize are biocompatible particles. These particles do not interfere the other nerve function. So we can safely assume that we can use this particle as a drug carrier or as a therapeutic particle for biomedical application. So with that, I told you about that how we made this particle, and then further characterization through the different technique, and then we went through the functionalization technique, and followed that we understand that how we deliver this particle to the brain, and then eventually we understand that how we detect this particle in the brain and their biodistribution. So with that, let me conclude the talk that we use electrospray technique to deliver this particle, single particle at a time, to the locust antenna, and we proved that these particles are reaching to the brain. The, the second thing, once these particles reach to the brain, they do not interfere the electrophysiology of the brain, or they do not interfere with the nerve function of the brain. Then the important thing about this study, or we can say the implication of the study, is that where this study finally go, how we will use that. The important thing is, this is non-invasive technique. We did not pierce any cell. We just spray some particle on a nasal surface so that this particle can reach to the brain. So it will not only help as a non-invasive technique, but this technique will also allow to any patient which is old or a kid or a young, just spray the drug on the nose and this particle will go to the brain. So you do not need to go to the clinic, or you do not need expert clinician to deliver the drug to the body, because suppose you need an injection, you need to go to some sort of experienced person or some nurse or doctor, so they can give you the injection. So with that, I would like to thank my research collaborator, and also thank to these animals which I sacrificed in this study. And with that, I would like to take your questions.